Hey everyone. So for the people that don't know me, I'm Bonnie. I'm the Early Career Council co-lead. And I'm currently a postdoc at NASA JPL. But a lot of the work I'm going to talk about today was from my PhD, which was at the Australian Centre for Astrobiology at University of New South Wales in Sydney. A lot of people, obviously in this group, know about the ladder of life detection and why organic biosignatures are important. In fact, organic molecules, they make up a lot of rungs on the ladder of life detection. Like if you count what's organic and what's a rung in the ladder of life detection, it's like 8 rungs. It's more than half. But if we go to the next slide, we can see that a lot of these compounds, organic compounds that are actually more preservable and that are detectable through deep time are more on the suspicious biomaterials area of the ladder of life detection. And the molecules that give more information about the kinds of life forms that they might come from do not preserve as well through time. So if we go to the next slide, if we're thinking about ancient life sites on Earth and ancient life detection on Mars, we need to think about what kinds of molecules we might be able to detect from billions of years ago. So they can't just have specificity, they need to have preservability. So one of the great things about being Australian is that I get to live in the same country as the Dresser Formation, which is my favorite place on the Earth. So in the north of Western Australia, just at the top, which is like the equivalent of going from Miami to Seattle from Sydney, is where the Dresser Formation is. And this is a 3.5 billion year old rocks, which host the generally accepted oldest evidence for life on Earth. And so what's pretty cool about these rocks is there's actually a lot of fossilized hydrothermal environments there. So the picture on the bottom right, those are fossilized hydrothermal chert veins from a marine hydrothermal sequence. And the photo above them is a photo of a rock type called geyserite. And geyserite is pretty important because as a discovery in the Dresser Formation, because geyserite only forms at land-based hot springs. It forms from the exact action of a geyser splashing. And so the discovery of geyserite in the Dresser Formation in 2017 actually told us that there were land-based hot springs with life occupying them, which pushed back the discovery of life on land by billions of years, which has direct ramifications for thinking about how we might apply our search for early life on Earth to early life on Mars. And so if we go to the next slide, we can see that the rocks from the Dresser Formation, they're on the right. You can actually see a lot of instrument scientists from Perseverance and Mars 2020 there. That's from a field trip when they all came out in 2019 to visit the rocks. That was a field trip I got to organize, which was very awesome. So if we think about the types of life that we can detect on the early Earth, these are at a similar time evolutionarily to when Mars may have been habitable. And then on the next slide, so thinking about these land-based hot springs then, something that's really cool about them, I guess, is that there are a lot of really defined facies within land-based hot springs. So the kinds of organisms that live right near the vent, they have to be adapted to be able to live, up to 100 degrees Celsius. And then as you move further and further away from the vent, you have life forms that can exist in more ambient conditions and that don't need those kinds of adaptations. And something else that's really cool about land-based hot springs is, when the geyser shoots up, it's full of supersaturated silica. And as that water cools down, it instantly precipitates and it traps all of the organisms, sort of like a microbial Pompeii. So you can see here, this is like a Jurassic aged fly's eye. So that fly's eye is like 150 million years old, which is incredible. There's also a stem of a plant here, and then also a plant root. Thank you, Bradley, for your excellent slide animation. I really appreciate that. So then what happens in these environments when it comes to organic molecules? If they're preserving physical fossils so well, could they also preserve organic molecules through deep time? And that's really what my PhD looked at, these Phanerozoic hot springs as analogues to the Dresser Formation and what kinds of molecules we might find there that might be preserved back in deep time. So all of these hydrothermal environments, they're very dynamic and transient. And, what we see on the surface is only a snapshot of them, like an iceberg. There's most of what's going on in a hydrothermal environment is going on in the deep subsurface. And so you have these hot reservoirs that are constantly cooking everything that's going on. You've got water traveling from rocks below the surface, potentially bringing organic molecules up to the surface and then depositing them, contaminating the field sites. So this image on the bottom left, this is just showing a common biomarker, chronachiol, for instance. This is a biomarker for archaea. But you can see it's quite a complex, chunky boy, and that doesn't really preserve well through deep time. And so the kinds of molecules that preserve better through deep time are the kinds of things like phytane on the right, which is preserved from the side chain of chlorophyll. And the molecules that preserve through time have less biological information attached to them. So they've been less attractive in the research that thinks about organic biosignatures in hot springs. And so we know on Earth over time, the main thing that affects biomolecule preservation is the application of heat and pressure. This gets rid of, sorry, Heather. So this gets rid of a lot of biological specificity, and over time you get left with a bunch of like aromatic rings, and you can't really see any sort of molecule that was part of biological function. And so this is problematic because in a hydrothermal environment, you've got lots of heat going on all the time. It's way more than a traditional sedimentary environment. So you're artificially applying diagenesis to your hot springs as they go. So you have a compound in a hot spring, it might be altered more in 50 years than you have in 100,000 years in a regular sedimentary environment. And there's sort of this advanced diagenesis. So we saw this, I guess, on the next slide, when I started working in some Jurassic hot springs. And so you can see on the left, this is a sample we took from the vent. This is actually like a geyser breccia. And you can see there's a lot of acid dissolution going on in this petrographic images. On the right, that's a sample from the edge of the hot spring in the distal apron, and that's filled with a lot of plant fossils. And so what I did was I crushed these rocks up and I analyzed them on the GCMS. And in that top image, what I really just want you to take away is that the vertical axis is telling you about the nanograms of organics per gram of rock. And so you can see the sample that comes from the edge of the hot spring has so many more organics than the sample that comes from the vent because it's being less altered. And then if you look underneath, you can see the total ion chromatograms from the GCMS. And you can see the sample from the vent, it has a lower variety of compounds. It did have some hopanes in it, but they're not actually visible in the total ion chromatogram. Whereas if you look at the sample that comes from the edge of the hot spring, you can see there's carotanes, there's hopanes, there's stearines, they're all in huge abundance and they're visible immediately. And so that shows that there's a huge variation throughout the sinter apron of where you might be able to detect life if you're looking for it. And then on the next slide, you can also see that the kinds of things that we find are a little bit different. So on the left side in this chromatogram, you can see it's very noisy. There's not very good resolution because the abundance of molecules was so much less than on the right side where you have the distal apron. The other thing that's pretty cool is in the distal sample, you've got preservation of beta-beta hopanes, which are extremely unstable compounds. And so you only find them in something that hasn't been heated up very much. The other thing that I think is really cool here is that in the distal apron, you can see the S and R enantiomers of the hopanes. They're just the paired sequence that go through each of these different chromatograms. You can see that the R is much higher than the S. And that's because the R enantiomer is the biological one, whereas the S is the one that's stable through geologic time. And so you can see that's a very immature sample. The R is much higher than the S. And then if you look at the vent sample, you can see that the S and the R actually switch. Sometimes the R is higher, sometimes the S is higher. And that's indicating that there's mixed inputs of organics in that vent sample. And it's not, you're not only seeing like one type of organic that's indigenous to the sinter, you're actually seeing a lot of migrated compounds that have been differentially heated. And so in the next slide, we also saw this in New Zealand. So these are chromatograms of aromatic compounds, and you can look at the ratio of methylphenanthrenes to phenanthrene and methyl naphthalenes to naphthalene to tell how mature a sample is. And basically, we did this for a sample that is from the surface that's never been buried. And we were getting signals that told us it had been heated up to the mid-oil window, which shouldn't really be possible. And what we actually realized in this paper was that we were getting signals from temperatures that would have been about a bit below the subsurface, and that it was actually hydrocarbons being migrated and emplaced into the land-based hot spring. So when we're looking at these hot springs, we can't just think about contamination from the lab. We have to think about contamination from the field. So this picture here just shows that the sample, it came from the edge of a hot spring, and so it really shouldn't have had such matured organic signals in it. And that's just how pervasive that contamination can be. So then when we move on to the next slide, when we're thinking about the reliability of organic biosignatures in planetary environments, we need to think about three things, really. The preservation of the compound, like could it be preserved in the rock that we're looking at? Compound ambiguity, can we tell what the source of this organic molecule is? And the third thing is contamination, whether it's in the field or in the lab. And so one of the ways that we look at this in deep time is to look at Raman spectroscopy. So the D and the G bands that are shown here in this image, they're actually 5 bands masquerading as 2 bands, like 3 squirrels in a trench coat. And you can deconvolute them, and then you can work out what temperature your organic matter has been heated to. But there are some problems applying this to hydrothermal environments, because this calibration is built on slow maturation over time, whereas in a hydrothermal environment, you have like hot flash pyrolysis occurring. And so traditional sedimentary biomarker and organic proxies may not be as applicable to hydrothermal environments, because it's a completely different way of maturing your organics. And so, yes, exactly. This, I guess, illustrates my point that all of these organic molecule proxies are built on these slow burial rather than this flash heating. And so on the next slide, we know that this can be a problem when we're trying to evaluate contamination, because one of the ways we evaluate contamination, particularly in deep time, is the survivability of the compound. So this is from Jochen Brock's paper in 1999, where they thought that they had detected photosynthesis in the Archean. And so actually that wasn't the case. And you can see right here that those S and those R enantiomers of the hopanes that I was talking about earlier, they're almost equal. And that wouldn't happen in a really, really hot sample, like the samples from the Archean that have been heated by metamorphism. And so that's one of the ways that we can think about discarding contamination. And so Brock's also had discovered 2A-methylhopanes, which they thought at the time were a biomarker for cyanobacteria, which weren't. And so he really thought this must be contamination. So he worked for like 10 years to get out this next paper with Rasmussen et al. That was able to look at the biomarkers in the kerogen versus in the free fraction of organics and show that what they were seeing was contamination and not realistic. And then in 2015, they released a reappraisal of hydrocarbon biomarkers in Archean rocks based on a very comprehensive study that looked at all the methods of contamination. And this is really what we have to think about when we're looking in deep time. Is what we're seeing real? Is this really a compound that belongs in the context we're detecting it in? And so in the next slide, I think that basically what I wanted to say is that when we're thinking about the reliability of biomarkers, we need to think about site-specific alteration processes. And without assessing the context of the formation and the diagenetic processes that have occurred since the formation, we can't accurately evaluate biogenicity and syngeneity of more ambiguous compounds that you can't directly tell from biology. And this is directly relevant to Mars because all of the compounds that we've detected so far are not definitively biological compounds. So this has applications to early Earth and to Mars. And so that's the end of my talk. This is my acknowledgement slide for my lab back in Australia and for my lab at JPL. These are 4 different papers that I was on that tackle, that I either led or was second author on, that tackle this problem. If anyone's interested in talking about organics and hydrothermal alteration, it's all I think about day and night. So please come talk to me about it. Thank you.
Thank you, Bonnie. Great talk. I suggest we hold on questions until the end, so we have to make sure every speaker has time to present. So who's going to be next?
I can go next.
Let's go.
Do you have sharing controls, Taylor?
I will let you know. Yes. Can you see that?
Yep. Excellent.
Okay. Let me put this in slideshow. All right. If something isn't advancing or you can't hear me at some point, just let me know. Okay. So today, I'll be talking a little bit about some remote sensing work and in situ observations of these acidic saline lakes in Western Australia. That is what mainly my PhD has been over. This is some older stuff. I've been doing new stuff recently, but just for this, I'll give a little bit of a background. Also, I did a terrible job of introducing myself, but in case you don't know me, my name's Taylor Plattner. I'm a 5th year PhD student at Georgia Tech, working with Dr. Brittany Schmidt, who is at Cornell now. And to give you a little idea of the project that I'm on, mainly my work is funded by the Oceans Across Space and Time team, OST. And it's basically this ocean world's mission to Earth designed to address gaps in our understanding of habitability in extreme terrestrial environments. And so we're kind of using these field analogs you see on the right to guide upcoming ocean worlds missions to our solar system. So that could be contemporary, relic, or remnant ocean environments. And so some of these field analogs that we've been to is the Great Salt Plains, Oklahoma, the Bass Lakes in British Columbia, Canada, South Bay Saltworks in San Diego, and today what I'm going to be talking about is the Western Australia transient lakes. And mainly our team is made up of multidisciplinary team across multiple institutions. So we've got a lot of biologists, geochemists, and also people that are more interested in like habitability. And so like physical measurements or like a geologist as well. And so again, we're using these field analogs to look at how these different environments that are analogs to contemporary remnant and relic analogs, how they change through geologic time as they go through stages of freezing, wetting, and drying. So it's just a little bit of background, and then just to show you kind of like how we work in our analog lakes. This is here in the South Bay Saltworks all together. Okay, so what I'm going to be talking about today is, again, the Western Australia transient lakes. And here I'm just showing a map of all of our field sites. So we went to Western Australia, and this is just showing a portion of Western Australia, particularly the Illegarn Craton, where a bunch of these lakes are. They're across the whole Illegarn Craton, and there's like hundreds of thousands of these lakes. And we were able to sample about roughly 40 lakes. So it was a pretty crazy fieldwork expedition in August 2022. And we had about like 20 members in the field all together, working at times jointly and then at times separately. And we were able to do some really cool science from that. I'll come back to that map again when I'm going to talk to you guys about the remote sensing part, but just to give you an idea of what these lakes are like and why we even chose them in the first place. So this would be our analog, that is our relic ocean environment. So that's what relic or remnant. So these lakes are going through these wet, dry cycles over time, and that's why they're really interesting to us. And so the main reason why we chose these analog lakes in Western Australia is mainly due to their age and their composition. So these lakes sit on bedrock that's about the same age as the Noachian and Hesperian terrains on Mars. And they also have similar compositions that you see in the environment. So there's a lot of sulfates, clays, iron oxides, chlorides, various similar mineral assemblages that we've seen across the surface of Mars. And so it really makes these field sites a unique analog for Mars. But also the reason why they're particularly interesting is because they've gone through these wet dry cycles for thousands to millions of years. So these lakes are essentially just sitting on the Ilgorn Craton, and their main source of like to be replenished is from rainfall, and then they mainly just evaporate over time. So they're just continually going through these wet dry cycles over time. And they've gone through these again for thousands to millions of years. And so it's kind of like a window into possibly understanding whether Mars dried up all at once or whether it went through these cyclic wet dry cycles. So we don't really know that. This is kind of an analog to maybe potentially understand that better, because these lakes are actively going through these wet dry cycles, and we can understand better these like water rock interactions that are happening over time. Okay. The other reason why mainly this will be kind of short, but these are also interesting analogs, basically because, right now we have been learning what early Mars was like with various rovers that explored past water environments on Mars. And so from the various data that we've got back from the rovers, they've revealed lake beds that are geochemically complex. And we know this based on specific geologic features that we've seen kind of imaged here on the right. So we've got these hematite blueberries that are imaged by Opportunity, and then the cross bedding that is imaged by Curiosity. And I particularly think these analog lakes relate more to acidic conditions. So thinking more about the places that Opportunity imaged, and particularly these hematite blueberries are interesting because you can also form these like hematite nodules in these Western Australia lakes. So it's kind of like understanding these conditions, that these geochemistry conditions where you could have formed such features. And so it's Western Australia in particular, these lakes are interesting because, on the surface of Mars, we can only see these relic environments. So we can only really get at a rough idea of what the pH and salinity had to have been like. We don't know specifically what the pH or salinity was like in these environments. We don't know what the ion composition was, the water activity, all these things that contribute to habitability. And so these analogs are interesting in the fact that we could actively look at these environments that are going through these water rock interactions over time, and maybe how that could have affected biomarkers in these environments, and maybe understand where the best places to look for potential biomarkers are in these types of environments. So mainly my work is kind of, you can think of it as being modeled as a mock mission. This is kind of how our field work was, how we kind of planned it. But basically, I've done a lot of remote sensing work. So that's kind of like the orbital part of the mission. And then our lander part of the mission, or rover part of the mission, is actually going to these lakes in situ with a bunch of the in situ instruments that we had with us, such as a portable spectrometer, XRF, Raman, and a LIBS. And then our laboratory study is kind of like the sample return, which is all the stuff that we're doing now back at the lab. And so that's what you kind of see here on the right with all of these images. 1 is the remote sensing, or the orbital part. 2, 3, and 4 is kind of 2 is we're kind of assessing the area. 3, we found something that's particularly interesting and we're zooming in on that particular feature. And then 4 is actually doing our measurements in situ. And why would we want to try to connect these in situ observations with the remote sensing? Well, the main reason for that is to really give context to where these potential biomarkers are more likely to be found, and connect the features to the environment that they're found in. So really possibly trying to make connections with these analog environments to various places that you see on Mars, and make some connections to where we might think biomarkers are more likely to be found in those environments. So we could have more of a chance of getting a positive life detection result. And as Bonnie kind of showed, I also have looked at the ladder of life detection for my research as well. And okay, so here I am showing our field site. So the main field site, I don't think I think I cut it out, but our field site that we're showing is the F21 field site, and that is what I'll be focusing for the rest of this talk on. In particular, this area is interesting due to the fact that it has a bunch of weathered rock outcrops. And so that was interesting in the terms of like a remote sensing perspective. So we could actually detect that. Whereas most of these lakes kind of are just sandy, nothing's really interesting geologically is going on, but there are specific lakes in Western Australia that have these features. And I think it's really important in terms of like, we know that salts preserve biomarkers over long periods of time, but what else do you find these salts with? And so that's kind of what we're trying to do in these environments. And so basically all I'm showing is kind of these weathered rock outcrops in these environments, but there's also these diagenetic features such as these quartz veins and other reddish materials that we think could be jarosite. We're still doing analyses on these to verify what it is. But so far, we do know that in this environment, they're mainly kaolinite and quartz dominated terrain. So I think that's mainly what you're seeing in the weathered outcrop, but there are iron oxides as well in these environments. Okay, and then also showing you guys kind of in situ what the salt precipitation looks like. So this is what was interesting to me through these wet dry cycles. They precipitate out various salts such as mainly halite and gypsum. And so as they do this, they can preserve potential biomarkers over time. So that was another reason to focus on these lakes, in particular where you find these potential biomarkers. So if we see some lake that is like more acidic or more basic, like which lakes are you mainly finding these potential biomarkers being preserved in? And trying to connect that with remote sensing. And so here we're just showing these precipitation of salts. So you can have various ways that the salts precipitate out. And what wasn't really, I didn't think that this would really happen in this environment, but I thought when it was in a wet cycle, it would have all of its salts dissolved. But when we went in situ, I think some of these lakes had reached saturation over time. And so you have this thick salt bed that doesn't get dissolved at all. And that's what you're seeing in this figure 1 right here. And so this is this light area. That's where you're seeing the salt crust on the bottom. And then 5 and 6 are just zoomed in images of that. Okay, I'm going to skip over this kind of quickly just for the sake of time. But mainly what I'm going to be showing you next is the remote sensing that I've done so far. And the method that I've used is from Radwin and Bowen 2021, basically using their method to map halite and gypsum in these environments, but also kind of like the weathered outcrop. But I didn't use their method for that. Okay, so our F21 field site, which I did just show you, this is the map of our field sites and where this is in Western Australia. And so of that particular field site, this is again F21, but from an orbital view. So this is just showing color images. And again, there's a wet and a dry cycle, and particularly in Western Australia, local winter and local summer. Local summer is when their dry season, and local winter, particularly in the Illigorn Craton, is more wet. And so that's what August 8th is showing. And then January 3rd is showing a more dry stage. And then the red dots are our sampling sites here at our F21 field site. And then I'm basically, from the methodology, what I'm showing right here is a classification image of our August 8th date. So this is when we went there in situ, and we knew it was full of water. But we're also like looking and see if we can identify these weathered rock outcroppings around the lake. And so what you see in dark blue is water, and then in the surrounding terrain, the orange and cyan is our mixture of sand and weathered rock in this area. And for this, for the methodology that I used, I didn't do a band ratio for gypsum or halite because obviously you don't see anything in this stage because it's all flooded. You're not going to see any salts. So there's no salts visible during this wet cycle. However, when you're looking at this dry stage with the classification image, this yellow is going to be your salt pan, and then orange and cyan is your weathered mixture of weathered rock and sand. And then you do have an actual band ratio image for this. So again, I did halite and gypsum because we knew that these lakes precipitated out these particular salts. And I promise I'm almost done just for the sake of time. But what we did is mapped halite and gypsum. And basically what you're seeing is red is halite and green is gypsum. As you can see, there's no red, but there is yellow. And so that indicates a mixture of halite and gypsum. And then on the outcrop skirts of the actual salt bed, there is gypsum present. And so we looked at this the spectra for this. And obviously you're already probably thinking, well, you know, chlorides, you can only do thermal infrared. This is a different method that has actually looked with Landsat at halite. So it is published, and if you want to go look at it, I was able to reproduce their results. And then I've also applied their method here for identifying like wet halite and then gypsum. And so what I'm showing here on the left is their spectroscopy work for mixtures of gypsum and halite. And then on the right is showing my work. And so the yellow is that salt pan that I was showing. And then black is our in situ sample of that salt. And then red is that in situ feature resampled to Landsat 9 specifications. And so as you can see, it doesn't really look like a gypsum and halite mixture. It kind of does. But when I compared it to their wet halite spectra, it looked more like wet halite. And so basically just showing how similar these features are. So we really think that the salt that we saw in situ was a wet halite sample, and we're doing more analyses on that for XRD and XRF, and all of our salt samples are currently getting analyzed for XRD and XRF for further analysis to verify if it's a mixture or whether it's just halite. And so then this is also the outcropping, the weathered rock outcropping. So this is what the cyan and orange is showing. And so that's the spectrum from that particular area in the image. And then on the left is kind of just showing similar spectra. So what it looks like for the weathered rock outcropping, it doesn't, like our actual remote sensing for Landsat looks more like gypsum halite, but I don't think that's what it is. What I do think it is, and I'm skipping through this just to show you, what I think the weathered outcropping is kaolinite, but there's also salt induced on this spectrum that you see right here. And I say that because it doesn't look exactly like kaolinite. So this is what a kaolinite spectrum would look like, an in situ spectrum, and then like resampled to Landsat 9. So it's a little different. And the reason being is because there is salts induced on this sample that we have of the weathered rock. And so we have done XRD and XRF, and it does show a mixture. For just to wrap this up, we observed increased abundance of evaporative minerals visible in and around the lakes during local summer. So this is consistent with undergoing a seasonal dry cycle at this time. And we're further trying to understand these environmental history of the lakes and understand what is going on over time with these water rock interactions. We're further identifying these salts and rocks around our field sites, which is ongoing. And then we're further trying to disentangle these signals from salt pans, the large outcroppings of weathered rock and windblown salt from the lake, resulting in a spectrum from the area that is a mixture of various minerals. So really trying to see if there's any of these features that like iron oxides or anything that we could actually see from remote sensing, like any of the diagenetic veins, anything like that. We're trying to see if we connect that with remote sensing. And I will wrap it up there.
Awesome. Thank you, Taylor. Let's hold on questions until the end, and let's go with the next talk. Please keep an eye on the clock because we still have 2 more talks to go, and we are close to the top of the hour. So who's going to be next?
I think it'll be me, and I think I'm actually the last talk, if I'm not mistaken, Bonnie.
Oh, okay, great.
Yeah,
So-
that's correct. Just Osama left.
Yeah. And I am about to share. Can you guys see?
Yep.
All right. It's great to follow Taylor and Bonnie's presentations because I think I'm following along sort of thematically in the same area. So for those who don't know me, my name is Osama Ilian. I'm in my last month exactly of my PhD with Dr. Matt Schrank at Michigan State. And this is just going to be sort of a little bit of a 50,000-foot view of some of the work that I've been doing for my PhD over the past 5 or so years in an extremely volatile environment of Lost City hydrothermal vent system. And so I hope you get excited about it as I do. So this thing will let me, there we go. So sort of calibrate everybody again from that, I guess this is a 20,000-mile view. But when we look at Earth and we start to consider things about habitability and where life is, a lot of the things that we think of are measured in a very coarse sense. So things like water, we have the right temperature, we have seasonality, we have the right atmospheric combinations of gases and the right nutrient abundance. And so you look at this lush green planet and you think, damn, there's life. But when you turn off the light, if you're looking at humans at that instant of time that I guess this picture was taken, you see that life may not necessarily be homogeneously distributed. It's very heterogeneously distributed in these hotspots that are very sparse and in some places far and few in between. And so as a microbiologist, I like to use this picture to really highlight the point that microbial life is similarly dispersed despite that they outnumber most other life on planet Earth. So the study site that I am focused on is Lost City. For those who are not aware of what Lost City is, it is a serpentinite-hosted mid-Atlantic ridge hydrothermal vent. It was the first of its kind that was discovered in the early 2000s. It is a process of serpentinization that results in basically these carbonate chimneys that are formed from precipitations from hot calcium and magnesium-rich alkaline fluids that are actually somewhere around 200 to 300 degrees Celsius in the subsurface, purely as an exothermic reaction. And the fluids themselves, by the time they make it through the venting, chimneys are somewhere between 20 and 90 degrees Celsius and pHs of around 9 to 11. Through the site, there are lots of dense microbial biofilms. None have been cultured so far as representatives from the site. Some are upwards of, some samples could have upwards of 10 to the 9 cells per gram. So it's a really dense population in these systems. But as far as microbial ecology and function is concerned, much of what we know about the microbes working in the system are really speculative. And this is based on a lot of bulk analysis that have been done over the years, over the past 20 or so years at the site. And part of it is owing to the difficulty of sampling this diffusely venting site. You could see the biofilms hanging out there. We can't culture any of them, but also if we try to do any deeper analysis on individual cells or try to capture any of those cells, we're not really successful. And part of it is just the nature of the matrix and the nature of the biofilms that are there. So Lost City is very mineralogically predictable. We have a site that's dominated by 3 major minerals, aragonite, brucite, and calcite, and they form under very different and constrained conditions. And so part of what I have been trying to do is use these minerals as a tool to actually try to identify a little bit more about the microbial microenvironment. And hopefully that could tell us a little bit more about what nutrients and substrates these microbes are able to use in those microenvironments. And so given what we know about these different minerals, we can hopefully use them as a proxy for an evolving temperature regime or an evolving pH environment. And so that gives us essentially a gradient of conditions along which these microbes could exist. We have omics data, so we have DNA that was extracted from this information, from these sites. We have mineralogy data from those same samples. And so we now have a nice data set that we could attempt to do correlative analysis on. And in some instances, you could see like the graph that I'm showing here on the right, some of these minerals are perfectly inversely related, especially in the case of aragonite and brucite. which form under two very different conditions and represent two different histories over the lifetime of some of these vents. So again, sort of as a very broad overview of what some of the findings we're working on right now, or we're actually putting this in manuscript, so I'm being a little bit intentionally vague, but some of the findings we see is that we do actually see physiological differences along some of these mineralogical gradients. So if we pick aragonite-rich samples, which you would be able to find aragonite in early formation of these vents, we find that a lot of the physiologies are focused on reverse CCA cycles with long-Dahl pathways, methanogenesis, and a lot of trace metal sequestration and transporting genes. As the vents age, we expect to see more brucite-rich elements there, and we see the metabolism shift towards things like assimilatory sulfate reduction, nitrogen cycling, methane oxidation, but also a lot of the trace metal genes and enzymes are more related to iron sequestration, in addition to some of the other trace metals that we mentioned earlier. And then as the vents age and they become extinct, a lot of those venting channels and interfaces become infilled with calcite. The aragonite ends up transforming into calcite, and we get a lot more of the aerobic processes that we'd see almost anywhere else in the ocean. It's a less extreme environment. And I would argue it actually begins to resemble a lot more the marine sediment. And we see a lot of things, again, like sulfur cycling, hydrogen metabolism, which is what you would expect from a site in the marine subsurface as well. And we can actually use this gradient to begin to tease apart some of the most sensitive microbes to those changes in the mineralogy as a proxy for changes in that environment. And in this heat map, you could actually see that if we put the mineral weight percents below it, we can start to identify these different clusters of microbial taxa that are more associated with specific concentrations and organizations of this minerals than others. We can also do something super interesting. And so we can actually try to take those vent samples and try to map them for both their elemental heterogeneity and their mineral heterogeneity without mushing them up and basically diluting some of those signals. And I mentioned earlier that some of these samples are very fragile. If you take them immediately out of the ocean water, they'll dissolve, they'll liquefy. And so this has been something that we've worked on for a little bit, and we ended up coming up with the solution of embedding the samples in resin and preserving all the in situ information for collection, and then submitting those samples to this kind of spatial mapping. And so we begin to identify things that are very cool, like this mineral heterogeneity, for example, between brucite, which is this teal mineral, and aragonite, which is our red mineral, and a little bit of calcite. We see this nice sinuous organization of the venting channels and how the minerals are actually organized in a very structured and heterogeneous way within them. But we can also do microscopy, which is super interesting because we begin to see these networks of cells that are almost a lot of the times exclusively associated with some of these brucite interfaces. And the way these microbes are structured is also quite interesting because as opposed to just this giant glob of microbes that are associated with the venting interface, we actually see this very structured network of microbes that seem to be organized in a very, very specific way according to the mineralogy they're attached to. But also we could submit this to elemental analysis. And so if we take the same sample and we use the semi-destructive laser ablation ICP time of flight mass spec, we can begin to see hotspots of elemental distribution that may be relevant to why those microbes are organized in the way that they are. And so in this instance, I'm showing you some hotspots of zinc that we are actually able to correlate as being positively related to those microbial hotspots in different parts of the vent. So the areas where the arrows are the areas where we have the microscopy images showed here. And you could see those spots here are those DAPI stained cells that are fluorescing, and they're associated with some of these zinc hotspots. And so combining these three methods, we're calling it a multimodal analysis pipeline, we can provide a spatial context to where the chemistry is, the mineralogy is, and where the microbiology is. And that could give us a lot more information about the content and the context of the microbial microenvironment, rather just a bulk analysis measure of here are the concentrations of specific elements or minerals or geochemistries within specific sites. And we can really begin to define not just that there's context to this distribution in terms of microbes, but if you were to take a step back and try to analyze these distributions in terms of the history of the venting samples, we can begin to see a story begin to unfold as to how the conditions have changed as the vent has aged. So some of these vents could be upwards of 100,000, sometimes 200,000 years old. And enrichment in some of these trace metals, the lithologies of some of these minerals can tell us about not just the conditions at which we sampled them, but also the entire mineralization and precipitation history of that venting sample. And I would argue that enrichment of some of those microbial trace metal cofactors, like we saw earlier with the zinc, for example, here I'm showing you arsenic, cobalt, copper, uranium, molybdenum, and zinc, can actually give us an idea of where some of those microbial populations may have persisted, or where there could have been activities over the life history of that vent where microbes were there. And this might be a byproduct of those microbial activities as a microbial concentration of those cofactors may be a byproduct of their biological activity. And so we can begin to relate those patterns also to the microbial history within those vents. So if you take anything away from this sort of mini summary of what I've been working on, it's that heterogeneity is the norm in a lot of these sites. Previous modeling at Lost City showed that there was potential for these steep natural gradients to form in small areas. My favorite paper that was addressing this was by Philip Bask in 2007 that actually took a piece of this carbonate chimney after it was discovered, and they submitted it to computational modeling for increasing concentration of nucleotides. And you could see that in a small environment, I think this is only about 3 centimeters or so long, where you have warm convective flow that's flowing and you have thermodiffusion, you can actually get concentrations from 1x to 1000x over the length of this area. So imagine the heterogeneity of concentrations then when it comes to trace metals, the distribution of organics, or any other compound that, especially in a carbonate system, may be more amenable to advection and absorption in addition to biotic activity. We see that there's an interesting question of this pattern of heterogeneity, which needs to be further explored. And so this begs the question of, can we address this as maybe a potential biomarker for habitability or biosignatures? We know that no one molecule is going to be the tell-all signal for life, but we really hope that there is a story to be told about some of these patterns also possibly being together contextually a suitable biosignature or an indicator of habitability in some of these areas. And so some of those patterns and their relationships may be better indicators for things like habitable zones, even in small areas like brines or old extinct hydrothermal deposits, or if we're thinking in terms of future missions to Enceladus or any in situ sampling of those environments, we'd need a much more finer resolution to try to assess how those patterns change over the sampling area. So I think I was trying to talk a million miles an hour to stay in time. Here's all the folks that have tolerated me over the past couple of years, and folks that have given me money. And I will stop there and stop sharing.
Excellent. Thank you, Osama. I'll let Bradley stop the recording and the streaming.
Yeah, I'm on it, looking for the control. All right, one stop. Now wait for the other stop. So many controls. All right, both of them are now over.
Awesome.